Hello everyone. So in the last session, as you know, we studied about the inheritance of two genes. To understand the inheritance of two genes, we crossed taking two characters. So that concept is known as dihybrid cross. Whatever we did, we took the two characters and we crossed, and we take the ratio of phenotype and genotype. So these are all the things we did yesterday. Okay. So today I am going to repeat the same thing. I am going to do the dihybrid cross, but I am going to take the two characters. Two characters like yesterday I took the round and yellow characters. Seed of the shape of the seed and color of the seed, I took those two contrasting characters, but today I will take different or some other character to explain you the inheritance of two gene or dihybrid cross. So here in the parental generation, as I told you, I am going to repeat the thing. I am just changing the characters. Two contrasting character I am going to change. Okay. So what characters I am selecting here? Let me write that here. So the first contrasting character is position of flower. So this is one contrasting character. Here two traits are present. One is axial and one more is terminal. Axial and terminal, so these two comes under the position of flower. So axial is the dominant one, dominant trait, and this will be the recessive one. So this is the first character I am selecting to explain you the inheritance of two genes. The second character is color of the flower. Color of the flower, as you remember, while doing the contrasting characters, out of 7 we have studied all the 7 characters. So in that flower color was also there. So here the thing is, the color of the flower is, I will write the dominant, and one more is recessive. The dominant nature of the color flower is purple, and the recessive one will be the white. Purple and white. So the dominant trait, the purple color indicates the dominant trait and the white color indicates the recessive one. So we have selected the two characters here. Let me write that one. In the parental generation, we are going to write the axial and purple. When this type of plant cross, or when it get combined with the terminal and white color flower plant, what will happen in the next steps? We will see one by one. Axial purple and terminal white. So this is the parental generation. Remember, when we take the parental generation for the cross or for the hybrid, don't forget to write the pure line of this plant. Pure line in sense axial. I'm going to write the genotype here now. This will be the parental generation or phenotype of the parents, and here genotype I'm writing. For the axial, I'm going to write the letter A. So capital A, it should be in pair. So capital A, it indicates the genotype of the position of flower, axial trait. Okay. So now to the purple, I will write capital P, capital P. So P indicates the color of the flower, capital P, dominant gene of the flower. Here Now coming to the recessive characters, recessive traits. The capital A, here we have written capital A in sense here we have to write the small a and small p and small p. So that's all about the genotype of parents, which clearly indicates the homozygous condition. Why? We have not written like capital A and small a. If it is written like this, the parents were in the heterozygous condition. In the offsprings, we don't know how the offsprings will be in the F1 generation. Though we need a clear cut, clear cut offspring, we want to cross the pure hybrid. So in the parent generation, in the F1 generation itself or in the P1 generation itself, we have selected the homozygous parent. We will select the gametes now. The gametes, out of these two, take one, and out of this take one more gene, and here out of this A and P. So these will be the gametes of this parents. Now cross these gametes now. Okay, consider this as a male and that as a female. What will be in the F1 generation? The F1 generation offspring will be capital A, capital P, small p. Write the phenotype of this offspring. Axial, purple. All right. Because we have a dominant gene here. This dominant gene suppresses the recessive one. This dominant gene suppresses the recessive one. So this is the F1 generation of spring. Alright. So now we will move further. We are going to see what will happen when we self cross this F1 generation. That I will take here. So I will write the headline like F2 generation. F2 means second filial generation. Yes, F2 generation F2 parental generation will be like axial, terminal, axial purple when it get crossed with the one more axial purple. But these parents are in heterozygous condition, that you have to indicate in the genotype. Capital A, capital P, small p. It get crossed with the capital A, capital P, small p. Work on it. You will come to know how these offsprings came. Alright. So, now the thing is, from this parent we have to take the gametes. So, when we start writing the gametes, by the time you have to understand the law of independent assortment, because while writing the gametes itself will come to know, will better understand the law, what it states. I will write the definition of that. You just write down. The law states, when two pair of traits, I am writing the definition of the independent assortment, law of independent assortment. This is the third law of Mendel. Okay, law of dominance, law of segregation, and third one will be law of independent assortment. But law of segregation and law of dominance we came across when we completed the monohybrid cross. If we have understood the monohybrid cross, we will come to know, or we can define the law of segregation and law of dominance. But if we want to understand the law of independent assortment, we have to understand about the dihybrid cross. Okay. So, what it states? It states when two pair of traits, here when two pair of traits, this is one trait, this is one trait, this is one trait, this is one trait. Okay, capital A, this is one trait or character, this is one character, this is one character, and this is one character. It becomes two pair of characters or two pair of traits combined to hybrid. When they combined, when they self-pollinate, when they cross with one another, segregation of one pair of character, segregation of one pair of character is, segregation of one pair of character is independent. Independent. I'll explain you how it get independently assort itself in the by the time of gamete formation. I'll explain you that one. So, character is independent of the other character, of the other pair of character in the time of gamete formation. Just complete the sentence. I don't have the place at all. I am going to erase this one because I have to write the Punnett square here. Okay. So, what we studied in the last class, we studied or we wrote the gametes. How we wrote the gametes? 50% of the gamete take the dominant gene of the one trait. So, this is one trait, one contrasting character, trait or character, both are similar. This is one contrasting character, this is one contrasting character. In the contrasting character, this will be the dominant and this will be the recessive. So, contrasting character we used to say. So, out of these two, this will be one dominant gene, in these two contrasting character, this will be the recessive one, as you know. So, here at the time of gamete formation, 50% of the gametes takes the dominant gene of one contrasting character, and so meantime this gene independently assort itself in the next in the time of gamete formation. So, how it get assorting itself? See, capital A, it came here, one more, a few more gametes take the capital A in sense, few more gametes take the dominant gene, and here also this also should come, this recessive gene also it gets segregate at the time of gamete formation. How it will get segregate? Like this, few of the gamete takes the, what is this, recessive gene, few of the gametes takes the this recessive gene. Now, as we are in the inheritance of two gene, this is not about the one gene, or this is not about the one character, one contrasting character. We have selected the two character. Meantime, this gamete, what it is taking the dominant gene, that gamete has to take one gene from one more character. How it will take? Just write capital P here and one more capital P, small p, small p. So, this is how the dominant gene, it will get segregated to the gametes. So, now one more we have to write here. So, these are all the gametes of the parent. So, this is what we got from the parental generation, that the segregation or the what is this independent assortment, it itself says the law of independent assortment. Okay. So, we will see the Punnett square, or we will see the offsprings now. Same, everything will be remain same, but you have to write by putting the Punnett square. So, I will erase this also because I need some extra space to write that Punnett square. Put 16 blocks. Yes, and at the top of this column or square, write one of the gametes. So, what I will write here, I am going to write the gametes. Do not forget, we have taken other character. Which is far different from yesterday's concept. Only character we have changed, but everything remains constant. Now, keep on crossing the gametes. Whatever you got, you have to write the genotype and phenotype. Genotype and phenotype. At last, you have to write the phenotypic ratio and genotypic ratio. All right, we will see the what will be the phenotypic ratio of this character and genotypic ratio. I will write, just follow me. Though we have completed this yesterday itself, that is why I am writing, but you have to write yourself, do not copy the same. And at last you can check or you can compare the offspring, and you have to compare the ratio also, so that it will become thorough, because this will be for 5 mark. Okay, in inheritance of 2 gene or inheritance of 1 gene, they will ask for 5 mark. Okay. So, and it is easier concept, easy concept it is, do not lose your marks. Okay, I am going to write the phenotype here: axial, purple, axial, purple, axial, purple, axial, purple, and here also axial, purple, terminal, purple, axial, purple, terminal, purple, axial and purple, the same thing. Axial, here white color is appearing in this offspring because it doesn't have any dominant gene. Here also white color flower is going to appear, axial white, axial purple, terminal purple, terminal white, axial white, here terminal white. Okay, so to better understand the concept, I am going to draw one line here. Just concentrate that line. So right from this block, wherever I have drawn the line, those all are expressing the axial and purple color, axial position of flower and purple color flower. Just look at this line, this one, 1, 2, 3, 4, 5, 6, 7, 8, 9. So all the 9 offsprings will be having axial and purple color. 9 is to terminal purple. How many terminal purples are there? 1, 2, 3. 3. How many axial white are there? 1, 2, 3. How many terminal whites are there? Only 1. So the ratio remains constant. As I told you, whatever you take the character, the ratio remains constant, but you have to follow the right manner of writing the gametes. Then only the ratio will remain constant, or else it will get mixed up. Okay, you have to write the genotypic ratio. And the next thing is, after this, we are going to study the chromosomal theory of inheritance. I guess in the last classes, in the first chapter, introduction class, I told you some history of Mendel. Right, just go through it. When I start telling about the history of Mendel, if you know about that history, it will become easier, because that is also important in the chromosomal theory of inheritance, that history, and then his work, when he started his work, and why it get neglected, and who was Mendel. All those I am going to repeat one more time in the tomorrow session. So